Hello, dear students. Welcome again. I hope you all are doing wonderful at home. We have started the post-fertilization events, wherein we have discussed the first post-fertilization event, the endosperm development. So we said that endosperm is very much required for the embryo development to have the assured nutrition. So we are going to discuss the next kind of post-fertilization event, that is embryo development. So embryo is forming from the zygote. We know the zygote is formed in the syngamy. Syngamy is fusion of male and female gametes in the fertilization. So this syngamy is leading to formation of zygote, and we know the zygote is present at the micropylar end of the embryo sac, and this zygote is developing to form the embryo. So that's what we are going to understand, how this embryo formation is occurring from the zygote in the embryo development. So embryo is developing at the micropylar end, as we have just said that, and because the zygote is present at the micropylar end, and this zygote is developing into embryo, and so embryo development is occurring at the micropylar end of the embryo sac. So the early stage of this embryo development is same in case of monocot and dicot, because angiosperm classification is, if you see the flowering plants, these are the angiosperms, and this angiosperm classification is into monocots and dicots. Monocot has one cotyledon, dicot has two cotyledons, and these cotyledons are uh, the basis of this monocotyledon and dicotyledon. The classification, we are going to understand them now. And this zygote develop only after certain amount of endosperm formed, because we know that endosperm is required for the embryo development, so that embryo gets enough amount of nutrition from the endosperm. And this zygote, so once it is ensuring that there is sufficient endosperm for the nutrition, they start developing further. So this zygote is first dividing into two cells. So what kind of division is occurring here? The division is usually mitotic division. So after the zygote formation, usually there are only mitotic divisions. Only during the gametogenesis, the meiotic division occurs. Otherwise, in the angiosperm plants, the embryo development includes only mitotic division. So here on mitotic division of zygote, they form two cells, and these two cells again they undergo one more mitotic division to form four cells, and these four cells are in a linear fashion. They are in the linear fashion like this, and out of these four cells these four cells out of these four cells, the terminal cell, this is like a filament, and in this the terminal cell is it is destined to form the proembryo, and the remaining cells which are present behind it, they are forming the suspensor cells. So the terminal cell of the filament acts as proembryo. So this terminal cell, this acting as a proembryo, the proembryo can be here or sometimes it can be even here. So it depends. The remaining cells of the filament, they are acting as a suspensor, and this suspensor, the function of this suspensor is they are pushing this this proembryo towards the endosperm, so that they get enough amount of nutrition from the endosperm. So the proembryo forms various stages of embryo development. So from here, this proembryo is forming into embryonic conditions, various embryonic developmental stages. So we are going to understand them through a picture that we are going to see. So first, before that, we understand the term proembryo. So something pro is referring to primitive here. So primitive or pre-designed, or you can say that this is an embryo at a very basic level, or it is in the initial stage. So that's why it is pro-embryo. So next, coming to the diagram, in this diagram, if you see here in this diagram, it is very evident. We can clearly understand from this. So the zygote, which is present at the micropylar end, and this zygote is developing to form proembryo in the filament stage, and this proembryo is undergoing various stages of embryonic development, like globular embryo, then heart-shaped embryo, and then finally into the mature embryo. So the embryo is in the form of like globe-like structure, whereas in the heart-shaped embryo, the embryo is in the form of heart shape, and the matured embryo, on the matured embryo, it is completely developed. It is having the two cotyledons. This is a dicot embryo. So we are going to understand the dicot embryo and monocot embryo clearly. I hope this picture is clearly representing the zygote development, and at least you get an idea how this zygote is developing into embryo here. So now we are going to see the two kinds of embryo, that is dicot embryo and monocot embryo. Starting with dicot embryo, the dicot is dicot. If you split the term di and cot, di is referring to two and cot is referring to cotyledons. So it is having two cotyledons. So the embryo has embryonal axis and it is having two cotyledons. These are the two parts of the dicot embryo. If you see the embryonal axis, so this this is called as embryonal axis. So this structure from here to here it is referred to as embryonal axis, and this embryonal axis is having the attachment of these two cotyledons, and the point of attachment above that, like the portion of embryonal axis, this is a portion of embryonal axis above the level of cotyledons attachment, above the level of these cotyledons is referred to as epicotyle. So I'll take a structure very clearly representing the epicotyle. So think this is a one cotyle, this is another cotyle. Cotyle is referring to cotyledon. So this is the embryonal axis. So this embryonal axis is, from this embryonal axis the two cotyledons are rising. So the embryonal axis above the attachment of these cotyledons is called as epicotyle. So this portion is called as epicotyle. So epicotyle is, and it is epicotyle. So the portion of embryonal axis above the level of cotyledons. So the portion of embryonal axis above the level of cotyledons is called as epicotyle. So here it is not very clearly distinct, but you can imagine this is considered to be epicotyle, and this epicotyle is terminating with the plumule. So this structure is plumule. So here also this structure, the terminating point of this epicotyle is plumule. So plumule is nothing but stem tip. Plumule is destined to form the stem tip, and this is leading to stem formation. So plumule is leading to stem, and the cylindrical portion below this point of attachment. So the cylindrical portion from here, so from here, so to the remaining part. So from here, the remaining part of embryonal axis is referred to as hypocotyle. So epi is above, hypo is lower. So just like epidermis and hypodermis, epicotyle, where it is a portion of embryonal axis above the level of this cotyledon attachment, and this is hypocotyle, where the embryonal axis below the level of these two cotyledons. And hypocotyle this hypocotyle is ending with the, it is terminating with the radical. Radical is nothing but stem tip, root tip. Radical is referring to root tip, and this root tip is covered again with the root cap. So this is a root cap, and this root cap is protecting the radical, the root. So from the radical there is root arising, from the plumule there is stem arising. So plumule leads to stem, radical arising to the roots. So this is a structure of dicot embryo, and this dicot embryo, we have to remember there are two cotyledons, there are embryonal axis, and this embryonal axis, it has epicotyle and it has hypocotyle, and this epicotyle is terminating with the plumule and hypocotyle is terminating with the radical, and this radical in turn they have root cap. So this briefly about the dicot embryo. We are going to see, so what are the examples of dicot embryo? Can you see this kind of structure in the dicot embryo? So usually if you take any seeds like gram, green peas or beans, or if you take the groundnut, any of these, if you just soak them in the water and just allow them to grow, you can, once you open them, you will be able to see all these parts and you can easily distinguish them. So going on to the next kind of embryo, that is monocot embryo. So in the monocot, mono is referring to one and cot is referring to cotyledon. So monocot is having only one cotyledon, and the rest of the part is like embryonal axis. It is same in case of dicot embryo and monocot embryo, and it has only one cotyledon, and that cotyledon is called as scutellum. is called as scutellum, and this scutellum is present on the side of this embryonal axis. So we are going to understand this. So at the end, at its lower end, it has radicle and root cap. So at the end of this embryonal axis, they have radicle and they are having the root cap. And this root cap in turn, it is having a covering by undifferentiated sheath. It is called as coleorhiza. Remember, rhiza is referring with the root. So coleorhiza. So this will help to remember coleorhiza. The other part is coleoptile. So these two terms are interchangeably confused usually, but we are going to understand them clearly. Once again, I'll erase this part. Yeah, correct. So here, this portion of embryonal axis above the level of attachment of this scutellum is again referred to as epicotyle. Same like in the Dicot, this portion where the cotyledon attachment, the portion of embryonal axis above the level of this cotyledon attachment is called as epicotyle. So this portion, entire of this portion is becoming the epicotyle, and this epicotyle has the shoot apex. So they are having the shoot apex and this shoot apex, and they are having the few leaf primordia. Leaf primordia means these are the structures which are going to be the leaf in the future. They are destined to be they are destined to be the leaf. So this epicotyle has, they are having the shoot apex and they are having the leaf primordia. All these are enclosed within a foliar structure. They are enclosed in a structure called as coleoptile. So this coleoptile is encompassing this covering the shoot tip, whereas this coleorhiza is covering the root tip. So coleoptile and coleorhiza, these two terms are again very important to understand. And the single cotyledon in the grass family is called as scutellum, and this scutellum is present on the side. It is present on the side of the embryonal axis. So this is the embryonal axis, and towards the side of the embryonal axis, they have this scutellum. So this scutellum is the only cotyledon that monocot embryo has. So this is a structure of monocot embryo. Example for the monocot embryo is like if you can take the rice and you can take the maize. These are all the monocots or the grasses. They're all the monocots, and this monocot has this kind of embryo. So we know embryo is developing from the zygote, and this embryo formation is seen both in monocot and dicot separately, but in the initial stages of this embryo development in the monocot and dicot is same, but at the maturation they are having very different structures. So these terminologies, all the terminologies of epicotyle, hypocotyle, scutellum, then coleoptile and coleorhiza and radical plumule, these are all very important. So I hope you have understood up to this.